Hi everyone, this is Purnima, Assistant Professor, Department of Botany, JSS College for Women, Saswatipuram, Mysuru. So here in the class we will discuss about a small concept called meiosis. So this meiosis process, it will be consist of some content. We call it introductions or definitions, mechanism, significance. So let us discuss by one. First, we will move on to the introductions or definitions of the meiosis. So in the year of the science in the year of 1905, the scientist called Farmer was coined the term meiosis process. So this meiosis process only in the case of the reproductive cells during the cell formations gamete formations. So during the gamete formations, the meiosis process only it will be done in the case of the reproductive cells, for example, egg cells and the sperm cells. And this meiosis process, it is called reductional divisions. Why it will be called reductional divisions? It means the diploid number of chromosomes, it will be completely reduced, or it will be formations of haploid number of chromosomes. So it will be completely diploid to haploid. So what you mean by the diploid and haploid? Diploid, it means 2n number of chromosomes in the organisms, or double set of the chromosomes in the case of the organisms. Again, haploid, it means in the organism which consists of a single set of chromosomes, that will be called haploid. So these reductional divisions, the meiosis process is a reductional divisions. The chromosomes number, it will be reduced from the diploid to haploid. So these diploid, it can be represented by the 2n. Again, the haploid can be represented by the n. So these meiosis process, the chromosomes number, it will be reduced from the diploid to haploid. That is the only reasons because meiosis is called reductional divisions. And these cells in which meiosis takes place are termed meiocytes or mother cells, sperm mother cells or egg mother cells. And finally, this meiosis process, which gives four daughter cells from the parental cells. So these parental cells will be produce four daughter cells. This is called introductions of the meiosis. And this meiosis process, it will be consist of the two divisions, that will be called heterotypic divisions and the homotypic divisions. So let us discuss by one. What do you mean by that heterotypic divisions and homotypic divisions? First, we can study about that heterotypic divisions. Hetero, it means what? Different. So these heterotypic divisions, it is also called first meiotic divisions. So in the first meiotic divisions, or during the first meiotic divisions, the diploid cells, it is divided into two haploid cells. So diploid cells, it is completely divided into two haploid cells. That will be called the process. It will be occur in the case of the first meiotic divisions. The diploid cells, you see, it is completely divided into two haploid cells. So then why heterotypic divisions? Why we call it heterotypic divisions? First meiotic divisions, why we call it heterotypic divisions? It means the daughter cells resulting from this division are different from the parental cells in the chromosomes number. So chromosomes number, it will be varied from parental to daughter cells. The daughter cells resulting from these divisions or meiosis process, then these different from the parental cells in the chromosomes number. That is the only reason the first meiotic divisions, we call it heterotypic divisions. So in the heterotypic divisions will be consist of the five types. We call it prophase first, metaphase first, anaphase first, telophase first, and the cytokinesis. Let us discuss by one. Prophase first. Prophase first, it is a longer durations. It will take some long time, or it will take some more time, because these prophase first will be consist of the five different substages. Five different substages. That is the only reason the prophase first, it has extremely longer duration as compared to mitotic prophase. So mitotic prophase or mitotic cell divisions, only it will be consist of the four stages that will be called prophase, metaphase, and telophase, and the cytokinesis. But here in the case of the meiosis, Then the prophase first, it will be consist of the 5 substages. We call it leptotene, zygotene, pachytene, diakinesis and diplotin. So let us discuss by one what you mean by that leptotene and what happens in the case of the leptotene. So all know about that, what you mean by that cells and what are the cell organelles. It will be done in the case of the cells. It means cell will be consist of the more number of cell organelles like that mitochondria, Golgi complex, then vacuoles, nucleus, like that. So here in the case of the nucleus will be consist of the nucleoplasm, nucleolus and the chromosome. So the whole happens in the only in the case of the chromosomes or chromosome structure and chromosomes variations. Then this meiosis process, it is only done in the case of the chromosomes. Then leptotene stage. So what happens in the case of the leptotene stage? The chromosomes become very distinct and long, uncoiled, and they occur in the diploid number. The chromosomes number is diploid, then it is very distinct, it is very clear, long, very long, and densely stained, again the uncoiled. And each chromosomes on the surface of the chromosome, that is the small bead-like structures, it will be appeared, that will be called chromomers. Every chromosomes on the surface of the chromosomes will be consist of the small bead-like structures, we call it the chromomers, and it is also consist of the centrioles. So I repeat, in the case of the leptotene stage, the chromosomes, it will be very long, very thin, and then lightly stained, uncoiled. Again, the chromosomes is diploid in number. They contain bead-like structure on the surface of the chromosomes. A small bead-like structure will be appeared, that will be called chromomers. Next, we will move on to the next stage called after leptotene, the zygotene will be happen. So what process or some of the what changes it will be occur in the case of the zygotene? So zygo, it means pairing. The two homologous chromosomes, that means one from paternal, one from maternal, or it is also called mother cells and what is also called father cells, and the two paternal cells, that will be called two homologous chromosomes, attract each other or approach each other, or pairing of two homologous chromosomes, that will be called zygote, or it is also called synapsis. Then these synapses will be occur due to the presence of the synaptomum complex. So what you mean by that zygotene? The two homologous chromosomes are after the leptotene, the zygotene will be appeared. Here in the case of the zygotene, two homologous chromosomes, that will be called one from maternal, one from paternal. Two homologous chromosomes, it will be attract each other or approach each other, or it will be interact each other, or sometimes the pairing of the two homologous chromosomes, that will be called synapses, and these synapses, it will be occur only in the case of the synaptomum or due to the presence of the synaptomum complex. Okay, so this is a zygotene process, it will be done after the leptotene stage. Two homologous chromosomes, it will be completely pairing each other, that is the process called synapsis, and this process or synapsis due to the presence of the synaptomum complex, that will be called zygotene process. Then here in the case of the zygotene, the chromosomes, it will be shortening, again the densely stained, again the coil. So in the case of the leptotene, the chromosomes, it will be very long, then thin, then also lightly stained. But here in the case of the zygotene, the chromosomes, uh, it will be shortening, then coiling, and also densely coiled. Next, we will move on to the next stage. We call it pachytene. Pachy, it means what? Thick or thick nature. So here in the case of the pachytene, so these are the stage after it will be done after the zygotene process. So this pachytene stage, two homologous chromosome of two individual chromosomes, it is also called bivalent. One from paternal, one from maternal, bivalent, and these bivalents, it will be completely split longitudinally. It means one paternal chromosomes, it will be completely split longitudinally. It will be a formations of the two sister chromatids, as well as in the case of the paternal chromosomes, one from maternal, one from paternal. So these are occurs in the both. Maternal chromosomes also split into longitudinally. Again, the paternal chromosomes also split into longitudinally. So both are split into longitudinally, and formations of the forming of tetrad stage. It means four chromatids will be formed. Sorry. Two chromosomes will be split, complete each other. Formations of the four chromatids. Then these four chromatids, it will be fuse each other, then formations of the tetrad stage. Next, very important stage, that will be called diplotin. That means what? So this diplotin stage, it will be occurred after the formations of the packeting. So here in the case of the diplotin, the homologous chromosomes, it will completely repel each other or retract each other. The two homologous chromosomes that separate from each other. Then in between of the chromatids, the segments or exchange of the genetic materials, it is nothing but crossing over. So in the case of the diplotin, the crossing overs will be happen. So what do you mean by that crossing over? In the case of the diplotin, the crossing over will be happen. It means what? Genetic segments will be exchange each other, paternal also and maternal also. Formations of the two chromatids, in between of the two chromatids, the exchange of the genetic materials, that will be called crossing over. So these exchange of the genetic material at some point, that will be called chiasmata. So it will be formations of the X shape, Y shape, 0 or S. So it will be completely differ in the shape. So it is also done by the X, Y, S and O, and also yes shape. The point, it will be crossing over, or the exchange of the genetic materials or crossing over, it will be occurred. The point, it is also called chiasmata. So the chiasmata, it will be happens more than 1, 2, 3, 4 like that. So more than chiasmata will be formed. So more than chiasmata will be formed, then percentage of the crossing overs will be higher. So in the case of the diplotin stage, the chiasmata will be formed, exchange of the genetic materials in between of the sister chromatids, that will be called crossing over. So due to the crossing over, the exchange of the genetic materials will be occur. Then exchange of the genetic character, it is also occurred. That is the then what happens in the case of the diplotin stage. And final one, diakinesis. It is also called terminalization. So in the case of the diakinesis, the terminalization process, it will be occur or it will be complete in this stage. The bivalents, or it is also called sister chromatids, tend to repel each other and migrate the periphery, or it will be moved towards the peripheral region of the nuclear membrane. Then nucleolus will be disappearance, and the nuclear membrane also begins to disappear. Spindle fibers will be formations in the cytoplasm. So that is the some of the changes, it will be occur and it will be moved towards the metaphase stage. So what will be occur in the case of the diakinesis? Terminalizations, it will be occur. So it means what? The crossing over segments or the bivalent segments, it will be completely repel each other and it will be moved towards the nuclear membrane. That is the towards the nuclear membrane. Then it is also formations of the uh, spindle fibers and the disappearance of the nuclear membrane. That is also called diakinesis. Then some of the changes, it is also called metaphase formations. So these metaphase formations, it may occur. So it means what? Complete disappearance of the nuclear membrane and the appearance of the spindle fibers. Next, we will move on to the metaphase first. Meta means middle. Okay, so this is the meiotic process. Process, metaphase first. So in the case of the cell nucleus, the all the spindle fibers are well developed and the chromosomes move towards the equator. The chromosomes, in the case of the diakinesis, the chromosomes will be moved towards the nuclear membrane, but metaphase first, the chromosomes all are moved towards the middle of the cell. So the chromosomes, it will be moved towards the middle of the cells. That will be called metaphase first. And the spindle fibers, it is completely well developed. Then also disappearance of the nuclear membrane. So in the case of the metaphase first, nuclear membrane completely disappeared. Well developed spindle fibers will be formed. Again the chromosomes will move towards the middle stage of the or middle point of the cell or equator. Then the two chromatids of the each chromosomes do not separate in meiosis and the centromere does not divide. Reminder, the chromosomes, it will be completely tied between the centriole. In the first meiosis or metaphase first or meiosis first, the centriole cannot be separate or the centromere cannot be separate or cannot be divided. Only the chromosomes, it will be occur. It will be move towards the equator. So then it will be formations of the anaphase. The centriole cannot be divide. That is the some of the most important changes in the case of the metaphase first. So metaphase first, the spindle fibers will be well formed, nuclear membrane completely disappearance, and all the chromatid materials or chromosomes materials, it will be moved towards the equators. Then we will move on to the anaphase first. So in the case of the anaphase first, each homologous chromosomes which its two chromatids, it is undivided centromere. So reminder it, the chromosomes, it will be moved towards the anaphase. The chromosomes, it will be moved towards the polar regions. Ana, it means polar regions. The chromosomes will be moved towards the polar regions. Then the only thing, the chromosomes will be moved towards the polar regions, not the centromere or centriole. So this is cannot be undivided centromere, move towards the polar regions. So in the case of the anaphase first, each homologous chromosomes, it will be moved towards the polar regions, cannot be centromere. Then this is the stage, we call it the dyad stage. Next, we will move on to the next stage, we call it the telophase, again the cytokinesis first. So telophase first. So here in the case of the telophase first, the haploid number of chromosomes will be formed. Then formations of the nuclei, nucleolus, then the nuclear membrane, disappearance of the spindle fibers. So here in the case of the anaphase, the chromosomes will be moved towards the polar regions. Then due to the formations of the polar region chromosomes, again the formations of the nuclear membrane, nucleolus, then the nucleolus. Then coming to the cytokinesis. So cytokinesis, it means what? After karyokinesis, karyo, it means what? Nucleus. Nucleus chromosomes will be divided. After divisions of the chromosomes, the cytoplasm also divided. So after the karyokinesis, the cytokinesis occurs. It means what? Divisions of the cytoplasm. Two haploid cells are formed. In many cases, the daughter cells pass through the short resting phase, that is called interphase. In many cases, the changes of the telophase may not occur directly, leads the prophase of the homolytic divisions. So sometimes it will be take some time, that will be called interphase or the some period. In some cases, cannot take the interphase process. See the whole pictures of the meiosis first. So meiosis first, it will be have the four stages that we call prophase first, metaphase first, anaphase first, and the telophase first. So prophase first, it will be consist of the leptotin, zygotin, pakitin, diakinesis, and the diplotin. So leptotin, in the case of the leptotin, the chromosomes, it will be very thin, long, distinct, and the lightly stained. On the surface of the chromosomes, the small bead-like structures, it will be appeared, that we call prophase or the leptotin stage. Then in the case of the zygotin, the chromosomes, two homologous chromosomes, it will be pairing each other, we call synapsis due to the synaptomum complex. Next we'll move on to the packetine. So each homologous chromosomes formations of the bivalent. So each homologous chromosomes split longitudinally, then formations of the tetrad stage. So after tetrad stage, in between of the two sister chromatids, the exchange of the genetic material that we call crossing over, then formations of the chiasmata. So in the case of the diplotin, chiasmata formations will be formed. Chiasmata, it means what? The point where it will be occur. The crossing over where it will be occur, the point we call chiasmata. Then formations of the chiasmata, exchange of the genetic materials and the crossing over vectors will be occur in the case of the diplotin. Then diakinesis. The all the chromosomes, it will be moved towards the nuclear membrane, that is called diakinesis, or it is also called terminalization. So this is about the prophase first. So after prophase first, next we will move on to the metaphase first. So all the chromosomes, it will be moved towards the equator region. So chromosomes, it will be moved towards the equator regions, that we call metaphase first. Meta, it means middle. So after metaphase first, anaphase first, it will be lead. So ana means polar regions. So anaphase, in the case of the anaphase, all the chromosomes, it will be moved towards the polar region in equally, then cannot divide the centromere. There is a small change or small information. So only the chromosomes will be moved towards the polar regions, not centromere. Next, anaphase will be moved on to the telophase first and the cytokinesis. So here in the case of the telophase first, the chromosomes will be uncoiled, then thin, then the distinct, then the each formations of the two gametes, the nuclear membrane will be formed, nucleolus will be formed, then the cytokinesis will be formed. So after karyokinesis or divisions of the karyokinesis, the cytoplasm, it is also divide each other. So here in the case of the first meiotic cells, two daughter cells will be formed. So next we will move on to the homolytic divisions. So homolytic division, it is also called second meiotic divisions, or it is also called mitosis process. So homo, it means same. There is no changes, it will be occur. So homo, it means same. So next, or meiosis 2. So in the case of the meiosis 2, the homolytic divisions will be occur. It is also called second meiotic divisions. Here in the case of the second meiotic division, there is no changes. Somehow there is no changes, it will be occur. It is same as it is the mitosis process. So in the case of the two mitotic divisions, during the divisions of the two haploid cells, formations of the during the first meiotic cells into two haploid cells. Two haploid cells will be formations of the four haploid cells, that we call homolytic divisions. So these divisions is called mitotic divisions, or it is also called second meiotic divisions. So these mitotic division, it is also called second meiotic division. Then why? It means two haploid cells will be formations of the four haploid cells, that will be called homolytic divisions or the mitotic divisions. The chromosomes, the daughter cells has similar to parental to the chromosomes number. The chromosomes number will be cannot be changed, as it is same in the case of the parental cell, also daughter cells. That is called homolytic divisions. And these homolytic divisions will be consist of the 5 stages called prophase 2, metaphase 2, anaphase 2, cytokinesis 2. So let us discuss by one. Prophase 2. So here in the case of the prophase 1, the chromosome changes, it is only in the one cell. But here in the case of the prophase 2, the changes, it will be occur in the case of the two cells, two haploid cells. So finally, two haploid cells formations of the four haploid cells. So the process, all the process that we call meiosis second process, that we've done in the case of the two cells. So prophase 2. So in the case of the prophase 2, the chromosomes appear distinct with the two chromatids and the very thin in manner. Then nuclear membrane will be formed. Then nucleolus will be formed. So in the case of metaphase 2, or it will be leads to formations of the metaphase. The all the chromosomes, it will move towards the equator region. Then the spindle fibers will be formed. So end of the prophase 2, the spindle fibers will be formed. Then the nuclear membrane will be disappeared. Then the nucleolus will be disappeared. So this is the prophase 2. So after interphase, the prophase 2 will be formed. So it is also done in the two cells. So in the case of the prophase 2, the diploids or the chromosomes will be very long, distinct, uncoiled, lightly stained. At the end of the prophase 2, the formations of the spindle fibers and also disappearance of the nuclear membrane and the nucleolus. Next, we will move on to the metaphase 2. Meta means also called middle. So all the chromosomes will be get arranged in the equator. All the chromosomes move towards the equator regions. Okay, so these equator regions or the metaphase 2, the centromere also divide. That is the only changes from the metaphase 1 and the metaphase 2. Here in the case of the metaphase 1, centromere cannot be divided. In the case of the metaphase 2, centromere will be completely divided. This is the only change between the metaphase 1 and the metaphase 2. So metaphase 2, in the case of the metaphase 2, all the chromosomes will be moved towards the equator regions that we call metaphase 2. Then the spindle fibers, it will completely well developed, then the attached to the centromere. Next, we'll move on to the anaphase 2. Ana, it means what? Polar regions. So all the chromosomes, it will be completely separated, then move towards the polar regions. So here in the case of the anaphase 2, the centromere will be completely divided. Then each sister chromatids will be moved towards the polar regions. That we call anaphase 2. Next, telophase 2. So here in the case of the telophase 2, during this stage, the daughter chromosomes uncoiled, nuclear membrane will be formed, nucleolus will be formed, then the nucleole will be reappearance. So it means what? 4 daughter cell will be formed. So in the telophase 2, 4 daughter cells will be formed, then nuclear membrane will be appeared, then nucleolus will be reappeared in the case of the telophase 2. Then these telophase 2, it will be occurred. Cytokinesis 2, it means what? Cytoplasm divisions will be occur. So after karyokinesis, cytokinesis will be formed. Then the two nuclear divisions and the 2 cells will be formed after daughter cells, resulting in the formations of the 4 haploid cells. So that is called cytokinesis 2. So after karyokinesis or after nucleus divisions, then the formations of the cytoplasm divisions. That is called 2 haploid cells will be formations of the 4 haploid cells, that will be called in the case of the meiosis 2. Then this is the whole pictures of the prophase 2. What happens in the case of the prophase 2? So as usual, as the prophase 2 is completely as well as the mitosis process. So mitosis process is completely covered the prophase, metaphase, anaphase and telophase, same as it is in the case of the meiosis 2. So these meiosis 2, it will be consist of the 4 stages. We call it the prophase 2, metaphase 2, anaphase 2 and telophase 2. So here in the case of the prophase 2, so it is the formations of the haploid interface. The chromosomes will be uncoiled, very thin, again the lightly stained. At the end of the prophase 2, formations of the nuclear membrane and the nucleolus. Then these leads to the formations of the metaphase 2. So all the chromosomes will move towards the equator regions and the formations of the what? Spindle fibers. Then these spindle fibers, it will completely hold the chromosome of the centromere. Then metaphase 2 will be formed. The next phase we call it the anaphase. So here in the case of the chromosomes will be moved towards the polar regions, then completely separate the centromere. Centromere will be completely divided and all the chromosomes will move on to the polar regions. Then telophase 2. So this is the last stage of the meiosis process. So here in the case of the meiosis process, the 4 daughter cells will be formed. Then all the chromosomes completely in the surrounded in the each cell. Then in the each cell will be consist of the chromosomes. Then it will be very stained, lightly stained, uncoiled in the very long end manner. So 2 haploid cells will be formations of the 4 haploid cells. It is also called reductional divisions and also homotypic divisions. So this is about the what happens in the case of the slide. So from the heterotypic division to the homotypic division. So prophase first, metaphase first, anaphase first and the telophase first. Next prophase 2, metaphase 2, anaphase 2, again the telophase 2. So from the prophase 1, diploid cell will be formed. From the telophase 2, 4 haploid cell will be formed. Diploid cell will be formed with the 2 haploid cell in the case of the meiosis first. Again, meiosis second, two haploid cells will be formations of the four haploid cells. That is called meiosis 2. So finally, what is the significance of the meiosis? So what process will be formed? So let us discuss by one. So gametes will be produced by the meiosis. So there is the only process meiosis. So these meiosis occurs, daughter cell will be formed. There is no meiosis, there is no daughter cells or gametes. So gamete formations, it will be only through the meiosis process. Then the constant number of the chromosomes in a given species are maintained by the meiosis. Then owning of the crossing over of the heredity factors from the male and female parents get the mixed. These causes the genetic variations among the species, variations and the raw materials for the evolutions. There is no occurring of the crossing over, there is no genetic material exchanges, there is no mutations or there is no variations. If it is occur in the crossing over and the genetic materials, it leads the formations of the mutation and also variations. If it is not occur, there is no mutation and there is no variations. So next, if there is no meiosis, the chromosomes is doubled or quadrupled, would not result in the formations of the monocells or the abnormal forms. There is no crossing over, there is no genetic variations. So it is leads the formations of the abnormal forms. It is also called mutations process. Then law of segregations of law of independent assessment of the Mendel operates in meiosis. So meiosis process, it is completely based upon the law of segregation and the law of independent assessment. It is completely genetic process by the Mendel, Gregor Johann Mendel. So this is about the significance of the meiosis. So finally, I would like to conclude about this topic. So let us meiosis. What do you mean by that meiosis? It means it's a reductional divisions from the diploid cells to haploid cells. From the diploid cells to haploid cells, that is the process called meiosis. That is called reductional division. So these reductional divisions will be consist of the two type called homotypic and the heterotypic. So first one, heterotypic divisions will be consist of the four process called prophase, first metaphase, first telophase, first anaphase first. Again, homotypic division will be consist of the prophase 2, metaphase 2, anaphase 2 and then the telophase 2. So prophase first, it will be consist of the longer durations or it will have the some long time, then consist of the five substages. We call it the leptotene, zygote, packetine, diplotene and diagonal. In the case of the metaphase 2, metaphase 1, all the chromosomes will be moved towards the equator or middle stage of the cell. Again, anaphase, all the chromosomes will move towards the polar regions. And the telophase, all the nuclear cells will be formed. Again, same as it is in the case of the meiosis first. Again, the meiosis second, it is a as well as the mitosis process. It means what? Prophase second, metaphase second, anaphase second, again the telophase second. So the overall concept, formation, only formations of the gametes. If it is occurs the crossing over, again the genetic material exchange, chiasma formations. If it is, there is no formations of the chiasma formations or exchange of the genetic materials, there is no formations of the new varieties. So it is abnormal formations or it leads the only the crossing, only the mutations forms and the abnormal forms. So thank you all. I hope all of you enjoying the topic or all of you understanding the topic. Thank you one and all.